just joining the recording. This is how your laboratory assignments will look like. They have been put on Moodle. So if you will go to Moodle, you will see an assignment given today as lab 1. There's a Google Doc link given, and this is what you should be seeing once you click on the Google Doc. Once you are here, please raise your hands so that I know.
Sir, we don't have access.
You don't have access?
No, sir.
I think. Okay, just give me a second.
Yes, sir. And now it will be access.
Can you try now? Okay, I can see. Good.
Yes, sir. Now.
Yeah. So what I would recommend doing is you click on file, you do a make a copy. Okay. When you make a copy, please save the file in this format BBL434 underscore lab 01 underscore your name. Let us say Ratnesh was the first one. Ratnesh, first your full name, Ratnesh Kumar Sharma underscore your ID. Okay, I don't know your ID, Ratnesh, so please forgive me. And then you can just have it as docx, right? So this is how you save the file. Once you have done that, go through this document. I'll go through it with you. Um, Once everybody raises their hands, then I'll know. Only 2 people have raised their hands, but I can see many more people have at least 8 people have logged into this. So I'll wait. I'll wait for 2 minutes until everybody has raised their hands. So meanwhile the people who have clicked on this, kindly please go through the learning objective today. So the goals is a short introduction to NCBI and other relevant databases. So this is basically a database for most biological information that we'll be working with in this course, and a short tutorial for retrieving information from these databases. And there are then 2 exercises after you have looked at this database. One exercise is this one called BRCA and the Angelina Jolie effect or Angelina effect. And the other one is basics of genes and IDs. So I still can see that only 10 people are viewers here. 11 people have raised their hands. I'm waiting for more people. Okay, so I think the numbers aren't changing, so it's saturated. So I'll begin. So basically today's goal would be it's just a beginning, is introduction to NCBI, that's National Center for Biological Information. That's a compendium of websites which are or compendium of servers which are storing different kinds of biological information. A lot of databases can be accessed through this, and a short tutorial based on different questions that can be answered using these databases. So the basic learning objective is to get acquainted with NCBI, Entres, Coffee Break, PubMed, Uniprot, PubMed, which is something that I introduced to you in the first class um, a, when a news article was shared about elephants not getting cancer. So this is the database in NCBI which stores all the publications. Then you have something called Uniprot, which is a database of protein sequences. Then there is something called RefSeq, which again is hosted within NCBI, which is a collection of a lot of DNA sequences that are there inside, collected across from lots of researchers. All right. So I am sorry that the TAs have not been able to join today. Everybody, please keep your hands down. I think numbers are not changing beyond 17, and you can raise your hands once you have a question. Okay. So I'm sorry that the TAs are not here. Both the proposed TAs are down with COVID, so they'll be joining us hopefully next week if they are recovering. Until then, you'll have to do with me. Okay, so please, the first task would be to go to each one of these websites, explore them a bit, just click around, just be comfortable. It might it might seem that it's a very like a redundant exercise or not something not very interesting, but you will be going to these websites again and again throughout the course. So it's very important that you get acquainted with these websites, and if you have any issues, just solve them in this class as soon as possible. All right. So, and after you have done that, then I'll introduce you to the question that is given here. I'm muting myself. If you have any doubts, Jatawat, you have a question? Or Jatawat Nayak? All right. Okay. Yeah. So please go through these websites. Uh, take 10-15 minutes. I'm here. If you have any questions, raise your hand and unmute yourself and talk, and I'll answer. All right.
Sir, we don't have here link for coffee break, sir.
So I've just received a request from somebody to edit the document that I've shared. As I showed you, please don't edit the document itself. Make a copy of it, save it locally, and then edit it when it will be required for you to edit it. All right, I hope everybody has briefly clicked around and looked at these different databases and links to them. So just to give you a revision of what's written here. So NCBI is basically a resource for different
We
kinds of
can
molecular
see your
biology.
screen. We cannot see your screen.
Yeah, I was not trying to show my screen, but I can show it now. Um, so NCBI is a national resource for molecular biology information, right? So it's basically hosting a lot of public databases that and conducts research in computational biology. A lot of software tools would be available through NCBI, as we will be seeing during the course of this course in various laboratories. And a lot of these tools are essentially working to understand molecular processes that affect human health and disease, and also looking at generally the all the other organisms out there. Next is Entrées. Entrées is an integrated information retrieval system for NCBI databases themselves. So Entrées, basically you can look at literature, so different kinds of papers, references. You can look at protein and nucleic acid sequences, so DNA and proteins, and even protein structures and population statistics in terms of how these protein structures might be differing between different populations. Um, this Entrée system essentially allows, it's more like a query. So it's like the interface, the first interface that you see to retrieve information from NCBI. Coffee Break is
Sir,
a collection of short reports on recent biological discoveries. Um, each report is basically also having different kinds of tutorials that show how to use different bioinformatics tools, and we would be referring to some of these during the course of labs. So PubMed is something that I've already introduced to you earlier. PubMed is basically the interface to access published research articles. And if you want to know anything about a research question or about some biological information rather than referring to newspaper articles, I would always recommend you to look at PubMed and try to get the exact uh, like a proper reference or a proper research study that could actually be explaining these things.
sir, when we are clicking at Entrées, it is directly going to PubMed and not Entrées. So, if you can clarify.
So that's good. So that basically means you have actually clicked on it. So Entrées used to be the previous version of NCBI to access specific things such as publications, but now it directly redirects to PubMed because that's the most relevant point, and Entrées and PubMed have basically been merged right now. So this was just to check whether you guys are actually clicking on things. Has anybody tried to click on Coffee Break yet?
Sir, coffee break, where is the
Sir,
link?
Have you tried to find it?
yes, sir.
Sir, on Google, I could.
Okay. So what's the latest article on Coffee Break?
Sir, where, like on Google?
Yeah, I mean, did you find the link of Coffee Break?
No, sir.
Okay,
No,
somebody did.
the Coffee Break is like a language learning podcast service on Spotify.
Yeah, I mean, if you go to Spotify, then probably you'll be seeing things on Spotify, right?
No, so that's
Uh,
like the first that we linked.
so if you do Coffee Break NCBI,
Oh, yes, sir.
so during the course of the lab, I'm also going to be having one of these trick things where I'll just be checking whether you guys are actually following what is being written. So NCBI Coffee Break was also an old version. It's an old thing. It has been replaced by something called Citable from different, from, for example, Nature. If somebody can tell me what is the latest article shown on NCBI Coffee Break, what was the date of that article?
Sir, October 20, 2014.
Sir, these are books or something like this.
Sir, October
Like, I
2024.
can't get exactly the coffee break.
Yeah. So,
So you were
sir,
not able to find a coffee break?
sir, I was able to find coffee break. The last article it says is from October 2014.
Okay. Can you share the link of coffee
It is
break?
not exactly coffee.
Yes, sir, I can
Yeah.
share it. On the Teams chat, sir?
Yes. All right. So Uniprot, I'll continue. So Uniprot is basically a database of protein sequences and functional information around protein sequences and how
This
they
is a
are.
book and not coffee break especially.
Were you not able to find coffee break?
Sir, it is a book link, and there are other links to, also on Google, which is referring to coffee break.
So I mean, it says right, coffee break tutorials for NCBI tutorials tools. I mean, the link might be something different, right? You have to read what's there written on
Yes,
the page.
yes, sir.
Yeah.
Yes, sir, that is there.
Yeah. So I mean, these are slightly old. I can maybe open and show it. So I mean, some of the things I hope you people will do yourself. These are like some self done exercises. But if you want me to click on everything and show you, then I can do that as well and waste time. Um, so see, the last one was of October 2014, so it's like a 7-year old thing. But a lot of these tools that I'll be mentioning, you can actually uh, click on one of these tutorials and actually look at this thing. So let's click on this one. So from the Statue of Liberty to the coin in the back pocket, what is the secret life of copper? So this is probably going to give you a tutorial on how copper is um, specifically found. So let's give some information about an organism, right? And here they'll be talking about a walkthrough the Entrez gene record for plasmin gene. So basically, if you'll follow this, you will be able to, they'll show you how to use NCBI to specifically uh, look at gene records. Something in the case of today's tutorial is very important because the second exercise is actually about looking at gene records and what kind of information that you would be having for a specific gene. Okay. So I'm going to go back to the tutorial now. So given that you have all this information of different databases. So for example, if you were to look at a publication, you want to search for a publication, you'll be looking at it in PubMed. If you want to search for a proteins information, proteins sequence, proteins function, you would go to Uniprot. If you want to look at the sequence, if you want to actually find the latest sequence or the letters of a gene, then you will go to RefSeq because has a collection of all the different kinds of genes that have been sequenced, so on and so forth. And Entrez is now been merged with PubMed. That's good. You have clicked on it, and NCBI has many other tools. This is basically some central location for repository for hosting all these tools. So now we'll go to the first exercise of the lab today. And it's sort of a pop culture exercise along with trying to understand how to find information about things that you probably would read in a newspaper or a magazine article. So um, a famous US actress, Angelina Jolie, she had what's called uh, a preventive mastectomy where she basically got a reconstructive breast surgery because she had mutations associated with she and her family actually had mutations associated with her getting breast cancer. So as a preventive measure, she got rid of the tissue that will get cancer, and that has led to a lot of people around the world actually being aware of the effects of this faulty gene called BRCA1. So the first set of questions that I would like you to explore using the databases that I've just introduced you to is some questions around BRCA1. So the first question is, can you find the first paper that was linked to BRCA, that linked BRCA1 to cancer? So this will help you to explore PubMed, how to actually look at papers, and how to sort of play around with the different settings in PubMed in terms of how to sort the papers by age and things like that. Next question is, find a paper which talks about a recent finding uh, with respect to BRCA1. Third question is, how many studies have been conducted in India about the BRCA1 gene specifically? And the fourth question is, does BRCA1 mutations only affect women? Or in other words, does BRCA1 mutation have effects on in men as well? So if you want to go deeper into this article, there are two articles associated with it, associated with the what's called the Angelina Jolie effect. These two articles are given here. First one is a more is a article by Cancer Research UK, which is a cancer organization, and this article is slightly more vetted than what you would probably read in a newspaper. And then the other one article is the effect of this preventive surgery and how it has changed the face of preventive cancer care. And this was published very recently in 2021. You can also go and look at this particular test uh, being offered by a very popular pathology lab in India, the Lalpath lab. You can go and click here and see how this test looks like if somebody wants to get it done and gets and basically understand how these things are probably done and how they are sort of being offered to you as a consumer of these services. All right. So I please go ahead and start to look into these questions. So the way that these assignments would work is for after each question, you can basically write your answer within this page that within this document that you have copied, and then at the end you can just submit the assignment in the Moodle in the assignment like in the corresponding assignment tab. All right, I am muting myself unless you have an immediate question I can answer.
Sir, I have a question.
Yes.
Sir, like if we search BRCA1 on PubMed, and again I search this term on Google Scholar, so why there is more number of papers on Google Scholar and
Good question.
yeah.
So Google Scholar not only has referenced publications, but it also has a lot of other sources from where it has articles. So Google Scholar also pulls articles from patents. Google Scholar pulls articles from a lot of public libraries where people must have given their thesis. So for example, when you will be doing your BTP1, all of you are supposed to submit a thesis. And typically these theses are actually, we are bound to put them online. That's why actually most of this work is checked for plagiarism. And once these things are put online, they might not be research papers, but they might be thesis, which would be searchable in this kinds of through Google Scholar, for example. So PubMed is
Sir,
usually just having articles, research articles that are published.
sir, all the thesis of PhD are the of our department or any other department at IIT are all these those published on online? Like I
Yes.
tried to search for that, but I cannot get that. I can get only few pages, few like contents and nothing more.
So I think they are supposed to be publicly available. I don't know the status of exactly how IIT like academics department does that, but I think most of the theses are supposed to be in public domain, and typically people like they do put it online. So yeah, maybe I can find out for you and I can get back to you about that. I can probably talk
Yes, sir.
about
Like
our
I
department how we do
see
it, but yeah.
only the table of contents is available and nothing more.
Okay, maybe you can if there is a particular thesis that you want to look at, maybe we can do that together after after this lab, right? You can send me the mess in a DM in a direct message, and I can work with you to access that thesis that you are interested in. Okay, Ratnesh, can you please keep your hand down unless you have more questions? Okay. So actually, let me do one thing. Let me just also quickly talk about the next module as well, because you can probably do both of them together. The first thing was just to get information about the BRCA1 gene in terms of the research that has been published around it. The second set of questions is around the gene itself. So you can essentially, the kind of questions that I would like you to answer is, uh, each gene is also having a unique identifier in a database, right? So you could probably, so in NCBI, each gene has a unique identifier. So I want you to find the gene's ID in the case of how it's stored in the NCBI database, which is very important because this helps you retrieve a lot of information about that gene, its sequence, so on and so forth. The next thing that I want you to find out is on what chromosome this gene is located on using NCBI, not using Google, please. The other thing that I want you to find out is about the gene's function. So based on the databases that I have talked about earlier in this lab, can you find out what is the product of this gene? What protein does it make? Where in the cell do you find this protein, and what is the function of this protein? Then specifically when you are describing the function of a gene or a protein, there is something called gene ontology. So each gene ontology is nothing but it's a description of a function in a more formalized fashion. So I would request you to basically for BRCA1 or BRCA2, look for the specific gene ontology associated with it, and then go to that gene ontology and understand what it's describing about the gene. And once you have done that, after you have answered these questions, maybe you can answer the final question that in your own words, can you explain what the BRCA1 gene does if you were to communicate this information to, say, um, somebody who is not, somebody who is understands biology and somebody who does not understand biology. So two kinds of descriptions, if you can write down after you have answered these questions. All right. So I am muting myself. If you have any question, you can ask me. And also I'm stopping the recording because I've gone through the assignment. So anybody who is not attending right now can basically answer the questions. So I'm here